Hello guys, we are from STB 1013 Cell Biology under Dr. Ho Wei Seng, and we are from Group 2 Subgroup 1 and going to present our video reports. This is a list of our group members: Audrey, Dan Ziqi, Elida Anak Bangka, Bernarda Anak Ambrus, Denny Hisham, and Virginia. And other from that, there's also Feliciana, Quatensi, Nathan, Jillian, Natalie, Nicole, and Dan Sunji. In this video, we will be sharing our journey and experience with scanning electron microscopy, or we also know it as SEM, during lab session. Through this experience, we have had the opportunity to take on significant responsibilities, work independently, and demonstrate strong leadership skills. We will show you how we approach challenges, make key decisions, and ensure successful outcomes, showcasing the quality of leadership, autonomy, and responsibility that have been central to each one of us. This practical had gathered a group of strangers where we were assigned to demonstrate the scanning electron microscope. We had set up a WhatsApp group to divide our tasks before the practical to ensure all the things came out well. After the practical session, we had face-to-face meetings between ourselves to divide the tasks. The member that played the role as leader, which led to our success. The guidance and the pros foster our collaboration, which ensure better outcome of our tasks. Next, the autonomy of scanning electron microscope. There is three parts of the autonomy which highlight importance and discussion. First, for the highlight part, group members can be seen discussing and making their own decisions on which part to do for the reports, and some group members make their own decisions to take videos for the group. Secondly, there is three importance. The first one is improve effectiveness of the research. Scientists can decide which topic is the most suitable for them to investigate, whether it is experienced or qualified. The next one is improve trust and satisfaction. Scientists are able to work independently without micromanagement. And the last one is have major breakthrough. Scientists are free to investigate any hypothesis without being controlled. Lastly, discussion part: how autonomy contributed to our group effectiveness. So the answer is it enabled group contributed members to choose the part that is most suitable for them. Next one, the lab report must finish in given amount of time with no problems. The last one is there's no conflict between our group members. Then we will move on to the responsibility when operating the scanning electron microscope. The first three responsibilities during the experiment are wear safety personal protective equipment, check the condition of scanning electron microscope before operate, and prepare sample with correct procedure. Then another three responsibilities during the experiment are operate scanning electron microscope with correct setting and avoid overuse, record reliable experiment data, and clean scanning electron microscope and environment after the experiment. So, how responsibility was upheld in tasks? First, we must ensure samples are properly prepared to avoid damage to the scanning electron microscope and specimen, and we need to strictly follow the manufacturer instruction for scanning electron microscope operation. Next, how responsibility was upheld in safety measure. First, we must follow all safety protocols when handling high voltage or chemical specimen, and we need to ensure scanning electron microscope is functioning properly before experiment and handling scanning electron microscope with care. Other from that, how responsibility was upheld in group collaboration. We need to share accurate results with others to avoid confusion between each other, and the novice should always work under supervision until proficient. Lastly, how the division of responsibility led to a group success. First one is increased efficiency, the second one is improved accuracy, the third one is next, for the reflection part, we had a cooperative and instructive experience using the scanning electron microscope lab. At high magnification, we investigated the fine details of sample surfaces, which helped us better understanding with the SAM technology. From sample preparation to data interpretation, each team member made a contribution that promoted collaboration and improved our analytical abilities. The significance of accuracy in scientific study was underscored by this lab. All in all, it was a worthwhile and interesting experiential learning opportunity. Then we will move on to the comparison of scanning electron microscope with other microscopy techniques. For the similarity of scanning electron microscopy with other microscopy techniques is they are all microscopy techniques, they are all instrumental techniques, and they are all used for both quantitative and qualitative analysis. And then for the method of analysis, all four of the microscopy are advanced method and microanalysis method. But for the instrument, scanning microscopy use scanning microscope, transmission electron microscopy use transmission electron microscope, scanning transmission in electron microscopy use scanning probe microscope, and atomic force microscopy also use scanning for microscope. For the energy source, scanning electron microscopy use electron beam, transmission electron microscopy use electron beam, scanning transmission electron microscopy also use electron beam, while atomic force microscopy use ions. Then for the type of interaction with samples, scanning microscopy, electron beam transfer a part of all of its energy to the solid which then emits backscatter and scattering electrons. And for transmission electron microscopy, electron beam pass through the specimen. And for the scanning transmission electron microscopy, at high voltage, an electric field at the tip of the cathode produces electron by quantum mechanical tunneling. The last one for the atomic force microscopy, the force acting between cantilever and sample surface cause deflection of cantilever following the tip which scan the sample surface. Lastly, for the applicable to the surface, there are scanning electron microscopy is conducting, transmitting electron microscopy is conducting, scanning transmission electron microscopy is also conducting, while for atomic force microscopy is both conducting and selecting.
Then, for the advantages of scanning microscopy, it has high resolution and depth, which allow 3D imaging of surface structures, and it has wide application for analyzing variety of substances such as metals, biological samples with coating and polymers. Next one is elemental analysis. It can reveal the elemental composition when used in conjunction with energy dispersive X-ray spectroscopy or Wilsonian test (EDS). Other from that, the limitation of scanning dermoscopy is it is not appropriate for moist or live samples because sample preparation need a vacuum and conductive coating and is very costly because of the expensive maintenance and purchase. Last one is image interpretation. Analysis requires expertise to interpret data accurately for effective comprehension of data. Lastly, the significance in scientific work. Scanning dermoscopy capacity to provide high-resolution images of surface and microstructures make it important in the fields of biotechnology and material science. The surface morphology of bacterial tissues or biomaterials is studied in biotechnology. Scanning dermoscope is used in material science to investigate the structural characteristics of coating polymers and nanomaterials. Lastly, the research and development depend on the ability to fully understand the smallest details, which can be enhanced by this technique. Next, for the conclusion part, participating in group activities during the scanning dermoscope demonstration allow for the development of leadership skills because the key initiative in discussion and decision-making processes showcase the importance of creative leadership in collaborative scientific endeavors. Autonomy enhance research effectiveness, foster trust and satisfaction, and allow for significant breakthrough as scientists can explore topics freely and it contribute to the group effectiveness by enabling members to select tasks suited to their skills, resulting in timely completion of the lab report with minimal issue and non-conflict. Other than that, by following the key of responsibilities, such as proper preparation, checking scanning international conditions before use, adherence to safety protocols, cleaning the scanning scope, and surrounding area after experiment, and accurate data recording will show a clear division of responsibility that enhance efficiency. Communication, skill development, collaboration, accuracy, and time management within the group.
Other from that, for the future implication part, the implication of this scanning electron microscopy experience extend beyond immediate academic benefits, as meaning advanced microscopy techniques position for future research opportunities potentially leading to roles in industry research or clinical settings. Moreover, the skill developed in leadership, autonomy, and responsibility will enhance employability and effectiveness in collaborative scientific projects. In conclusion, the scanning microscopy laboratory demonstration serves as an experience that enriches educational background while preparing for a successful career in science. The insight gained will not only inform the future studies but also equip you with essential skills that are applicable across various scientific disciplines. Lastly, we'll move on to the closing acknowledgement. To end our video report, we would like to summarize the key points of our scanning microscopy demonstration. This experience allowed us to demonstrate leadership, autonomy, and responsibility in scientific settings. Through leadership, we plan and manage tasks effectively. Autonomy was shown as we made independent decisions during the experiment, while responsibility was maintained by following safety protocols and working well as a team. This demonstration provides us with a valuable knowledge about the principles and application of scanning electron microscope in cell biology. It also highlighted the importance of teamwork and individual contribution, which will be beneficial for our studies and future careers. We would like to express our heartfelt gratitude to everyone who supported us throughout this demonstration. First, we sincerely thank our lecturer, Associate Professor Dr. Ho Wei Seng, for his encouragement and his time to be with us throughout our session. Next, we extend our gratitude to our lab instructor for teaching us the principle of scanning the microscope and guiding us in using the instrument effectively. Your clear instruction and support were invaluable. Finally, we thank our group members for their teamwork and dedication, which play a significant role in the success of this project. We also acknowledge the resources and material provided which made this demonstration possible. Lastly, we thank you our viewers for taking the time to watch our videos. We hope you found it helpful and insightful. Thank you for watching.